Hi, my dear students. Hope all of you are fine. I am Maria Joseph, Assistant Professor of Mar Basilis Christian College of Engineering and Technology, Pre-Med, leading this session on microscopes. Okay, last class that we have already studied about the scanning tunneling microscope, and now we are moving on to the another type of microscope. It is the atomic force microscope. Okay, it was actually developed as a modification of our scanning tunneling microscope for dielectric materials. Okay, now atomic and molecular forces are monitored and used for the characterization at the atomic scale. That is, atomic and molecular forces are actually noted down and are there, and it is used for the characterization of the studies of the atomic scale. Okay, forces are detected by a probe tip mounted on a flexible cantilever. We will see the figure, then we will understand how it is working. Understood? Deflection of cantilever is directly proportional to the acting force. As the name says, the deflection of the cantilever will be proportional to the force acting, and we will see the working of the microscope. An atomic force microscope is insensitive to the electronic structure of the sample, but a scanning tunneling microscope is not like this. Okay, this is the basic structure or how it looks like. An atomic force, this is the figure of an atomic force microscope. Now, it has got two modes. It is a widespread, it has got two modes mainly, that is the contact mode and the non-contact mode. As the name says, the contact mode, it is a widespread technique to obtain nanometer resolution images on a wide variety of surfaces. Okay, similarly, we have a non-contact method, that is, the tip moves 5 to 15 nanometer above the surface. Okay, this is used in situations where contact might alter the sample in subtle ways. That means if the contact can cause some problem in a sample, then we use the non-contact method. Okay, when the tip is positioned at the end of the cantilever, is attracted to or pushed away to the sample surface, it deflects the beam and the laser measures the deflection, which means the cantilever and the tip are positioned near the cantilever. This is the cantilever and the tip. Okay, the cantilever and the tip are positioned near the surface with a microscopic positioning device. Okay, fine. Now, the cantilever deflected with a photodiode that records the position of the laser beam that has been reflected off to the top of the cantilever. That means the cantilever deflected with a photodiode, that is, the photodiode is here. Okay, and we have the laser beam here. The cantilever deflected with a photodiode and it records the position of the laser beam that has already been deflected. Understood? The AFM then produces visible profile of even the small hills and valleys, and that makes up the sample surface. That is, it can measure even the small ups and downs in the samples, and it is found to be superior to the electron microscope, as we see every now and then. Okay, then next type of microscope is scanning electron microscope. Okay, It uses electrons rather than light to form the images. Understood? That is, it uses electrons rather than light to get some images. And it uses, as the name says, it uses a focused beam of high energy electrons to generate variety of signals. We'll see the figure. See, a scanning electron microscope consists of an electron gun. Electron beam is here, and here we have anode. Then we have two magnetic lenses. Then we have detectors and secondary electron detectors as well. And here we have stage, and here we place the specimen. Understood? So these are the parts of the scanning electron microscope. Then it produces image by probing specimen with a focused electron beam. That is, as we see, we focus the electron beam on the specimen. Okay. Then at each point, electron beam loses some energy. The lost energy is actually converted to some other forms like heat, or it can be like emission of secondary electrons, or it can be in form of light, or it can be in form of X-rays. Okay. Usually the image is constructed from signals produced by the secondary electron detector. Here we have a secondary electron detector, and usually the image is constructed from the signals produced by the secondary electron detector. Then we have the signals from the electron sample interactions reveals the information from the sample. That is, the signals from the electron sample interactions reveals the information about the sample, means it gives the information about the external texture or the chemical composition or the crystalline structure or orientation of materials that are making up the sample. It gives the information regarding the texture or composition of the signals. Okay. It displays the image corresponding to the position of the beam. Understood? That is, on the specimen where the signal was actually generated. These are the some of the properties of the scanning electron microscope. Now moving on to the advantages. To generate high resolution images of shapes of the objects and also to show the spatial variations in the chemical compositions. To generate high resolution images of the shapes of the objects, we basically use this type of microscopes to generate high resolution images and also to show the spatial variations in chemical composition. As we know, this is very important in the case of nanoelectronics. Now, characterization of solid materials. That is the name of the chapter, the characterization of materials. That is one of the advantages of scanning electron microscope. Now, large amount of sample is possible. That is, big samples are possible in the case of SE. Comparatively, larger sizes are possible. Then we have got high resolution. Then, preparation of the sample is very easy in the case of a scanning electron microscope. That is all about the functioning of this, and hope all of you will understand. Thank you.